Good morning. It's my pleasure to kick off the new year with an introduction of our guest speaker. Before I do that, a special thanks to Dr. Root for, it would take too long to thank him for all of the things I should thank him for, but thanks for mentorship for all of us, and for today, thanks for endowing this lecture series in trauma and critical care. Dr. Mayers, our visiting speaker today, he was born in Wheeling, West Virginia, went to Notre Dame for undergraduate, then went to Duke Medical School. Following medical school, did his internship at Parkland, but then followed those UT Southwestern Parkland surgery faculty to the University of Washington, where he did his residency. He did a postgraduate fellowship in immunopathology at Scripps, and he's been a world leader with respect to surgical translational science since that time. He spent his entire career at the University of Washington, starting in 1981, and he is the model and premier academic surgeon. He's the current president-elect of the American Surgical Association. He's been past president of the University Surgeons, the American Association for Surgery of Trauma, the Halstead Surgical Society. He's been a director and was chair of the American Board of Surgery. He has received two numerous awards to list here, but essentially a who's who of critical trauma and critical care surgeons and translational science awards. He is going to talk to us today about the response to injury and stress. It's my pleasure to introduce Dr. Mayer. Dr. Mayer.
Thank you, Ronnie. It's a pleasure to be here. As those of you who know Dr. Root, he made me promise last night that I wouldn't mention his name, but I'd already made the slides, plus I told him I lie like we all do. Because it is indeed a pleasure and honor to be able to give this lecture created by Dr. Root and named in his honor. Dr. Root has been a mentor and a supporter of us in this field, primarily of trauma. If I can push the right button. Which one is it?
That's usually used.
Oh, sorry. Okay.
Mm-hmm. That was not good. Hold on.
But Dr. Root is one of those gentlemen that you meet early in your career, even early in my career. And right, he's always there. He's always positive. He's always supportive. And he's been a mentor to many of us. He and Dr. Pruitt in the audience, who I have to also recognize, has been a lifetime mentor. He keeps me honest sometimes. But you have two phenomenal, long-standing leaders in this field. And you should be very proud of those people to be with you and actually get everything you can out of them while you can, because they're wonderful resources. As was mentioned, I did some work at Scripps in immunopathology looking at the aberrant inflammatory response. And the reason I did that is during my residency training, I was always fascinated why some people did so well in the ICU and other people did so poorly. Multiple organ failure, when I was training, had become a major focus of research. And so I went there to learn from a group of very bright PhDs on the cell biology of innate inflammation. I've sort of done that research for my entire career. I've been privileged to be funded by the NIH and DOD and other mechanisms for the last 30 years. And it's been a great intellectual challenge, and it's been fantastic. over the last 10 years, or 12 now, we put together a multi-center study. Myself, Dr. Tompkins was the PI from Harvard. We joined with the Stanford Genome Institute with Ron Davis, who did a lot of work on the original genome project. And we actually pulled together 7 trauma centers, and it was a true national, multi-institutional, multidisciplinary study, which is what the government now feels, and I think correctly so in many avenues, is the construct that you have to follow to actually pursue modern research. Doesn't mean you have to, but it's one that's becoming increasingly viable and one that does produce results that you just can't get in a single institution and not in a single laboratory anymore. Although again, there's a lot needs to be done in that arena. I think these approaches, which we ended up calling the glue grant because it glued together all of these different aspects of multidisciplinary groups, put together a lot of information. I just want to share some of that with you. So this is the old paradigm. This is the one that actually was put together not long after I became an attending in the early 80s. It was a theory that actually progressed to becoming pretty much a dogma. And so we sort of, once you get into the dogma stage, you no longer have to prove that it's really a theory anymore. You assume that it's correct, and therefore you force fit everything you see in your patients and everything you do in the laboratory to the theory rather than objectively observing what's going on. And it did fit much of what we saw clinically in the ICU. And that is that you have a stimulus, hypoperfusion, all the things that we were learning in the trauma field, ischemia, reperfusion, burn injury, soft tissue injury, fat embolization, and so forth would cause this a massive inflammatory response, and you would develop very rapidly multiple organ failure from bystander effect from this inflammatory response to the inciting stimulus. And then most of those patients would then come back towards normal slowly. Those who didn't, as is shown, do I have a pointer? Yeah. Those who don't return to normal up here persist with their multiple organ failure, which is fulminant, and they go into death. What happens, what we proposed is that the body, trying to restore homeostasis, would develop a counter-inflammatory response called CARS, and that this would then try and suppress the innate inflammation, return it to normal. In many people, there would be an over-response, and so of just coming back to normal, people would end up with a suppression of their immune system, and they couldn't fight infection anymore, which again fit the clinical picture of what we saw in the ICU. Those patients would develop secondary complications, secondary infections, ventilator-associated pneumonia, and then they would have a second hit. And then the second hit would again set off the inflammatory response, another episode of multiple organ failure, and this late death that Dr. Trunke described back in the early 80s, and you can argue about whether it really exists anymore, the timing and so forth. But basically, as you've heard from others, like PICS is now a disease that Fred Moore and others have created. Of these patients that you see in the ICU, they're there for 2 weeks, they have persistent multiple organ failure, they have multiple complications, and they go on to die from their secondary insults out here. And so this is what we live with. And it really did describe much of what we saw clinically in our critically injured ICU patients. So along those lines, these are the patients we're treating. This was back in the old days when we were drowning with salt water. Many people accused my old and original mentor, Dr. Carrico, of creating this saltwater drowning philosophy with Dr. Shires when he was in Dallas. And if you go back and read their papers, what they said is if you're critically ill, you need more fluid back during reperfusion than you did in original losses. And so that's where the 2-liter bolus came from and ATLS and so forth. But that's all they said. The rest of us took that and extrapolated the 2-liter bolus to 20 liters or death occurs, whichever happens first. And in that mind of the enemy of good being better, we'd give these patients 20, 30 liters of Ringer's lactate, and this was the end result. I mean, you can drown someone and keep them alive with a good ventilator and other support systems. Now the young residents never see this anymore. This greatest episode of iatrogenic injury in the trauma world by saltwater drowning has now been recognized, reversed, and we virtually never have open abdomens in the ICU anymore. If we do, they're for 24 hours until we can go back in a resuscitated, stable patient and do definitive gastric or intestinal resections, anastomoses, and so forth. But this, because of the abdominal compartment syndrome, is really not seen very often at all anymore. Although it's nice because it's one of the few examples that I can give in medicine where iatrogenesis created a disease, which was massive fluid overload, multiple organ failure, abdominal compartment syndrome, which then led to the formation of an international society to study the abdominal compartment syndrome and enabled people to create their academic careers and travel around the world based on iatrogenesis. So it did have some benefit to the doctors, but not much for the patients. So we learned that, and we quit doing that. So what else has changed? Well, back when Tompkins and I started writing this grant, I remember it was in the, if you know Dr. Tompkins, it was in a bar in South Beach at the Shock Society, which is an organization that does a lot of this type of research on multiple organ failure and outcomes of severe injury. And we sat down, we started sketching it out, and it took us about 3 years to get it funded, and then it was funded for 10 years. But at that time, what was being discovered are these major events outside the realm of surgery pretty much, and that was genomics and genomic technology. The Human Genome Project mapped the entire human gene for the first time. And even though it wasn't very usable, it did give us the construct of pretty much the entire human gene, a lot of its spliced variants, and allowed us for the first time to actually look at what the genomic pool in the human setting was doing in response to various diseases, and primarily it was focused on cancer. Secondly, there are other projects such as the Heatmap, which develop techniques of looking at, in this case, SNPs that tend to be bundled, and they tend to go together on sections of the human genome, which are the haplotype measurements that allow you to screen enormous numbers of aberrant gene expression in a very efficient fashion, and it allowed us to get a lot more information a lot more quickly about how the human genome was functioning. And then lastly, and this is where we were working with the Stanford Genome Institute, they and WashU and a couple other centers put together the high throughput technology that allowed for the rapid assessment of the entire human genomic response to any disease at any point in time from any tissue that you could extract it from. And it could be done, it was laborious, cost $5,000 a sample. That's why we went through a lot of money in this grant. But it was the first time ever that you could mass screen how the human body genetically or genomically was responding to an event. And so doing that allowed us to reconstruct the way we look at learning new information in biology. Sorry about the overlap at the bottom, but basically we had the microarrays, which I've just been discussing. At the same time, there was ways to mass analyze the proteomics, either through multi-screens or through mass spectroscopy, and you can look at the number of proteins being made. Now it's down to a single cell level. You can do separate cell separation, so you can see which cells we're doing alone and together. And then we started to develop the technology to actually analyze. One of the biggest hurdles which still remains is, as you'll see, we generate so much data. There's no technology to truly analyze the data accurately and quickly. And that's still a major hang-up we're dealing with. But with this information, then you can start to look at the basic cell molecular messaging and modeling, which is where I spent a lot of my time, and a lot of us did in this field in shock research, is looking at the various molecules inside the cell that control the cell, control the production of inflammatory mediators and so forth. And then you can start to look at the networks, the modules, the inflammatory module versus the other anti-inflammatory responses in the cell. And then you can start to look at the systems biology, looking at the overall innate and adaptive immune responses. And so building on this basic sort of like DNA level product and information, we can learn a lot about cell biology and systems biology. So this was our group. Again, Dr. Tompkins and a very close friend, Dr. Pruitt's, are burn physicians. And we had a lot of input from the, in Dr. Herndon's group just down the road. We had a lot of sampling and input from the burn community. And then the rest was from the trauma community, as shown here. There's many different ways, as I just showed you, you can work at it. We basically, what I'm going to show you is the data is from the far left side, which is the Buffy coat. We chose Buffy coat because we had access to it serially. Buffy coat from the circulation of these injured and burned patients. Now you could say, well, what's more important is the lung, the liver, the spleen, whatever. The problem is that the IRBs won't let you do serial splenic biopsies on your trauma patients. They may let you do one early on, a big one, but once you get past that point, it's kind of hard to do serial. And so it's just a reality. We were limited by what we could get our hands on, and it was a circulating coat. And the assumption is it may not be perfect, but a lot that's going on in the genomic response in the white cells is probably going on in other tissues, although the specifics have to be tracked down rather than just assumed that they are. And then you can see at the top there is the overall response, and then you can take each cell population again and start to break it down into the different cell types rather than just lumping. And we ended up with the burn patients and trauma patients, as you see. So this is it. We had 1,600 trauma patients screened to get the genomic information. One of the things that was nice out of having this money was it enabled me and some of the other trauma centers to push very hard is that we collected probably the most in-depth physiologic database on severely injured trauma patients that the world's ever seen, because we wanted detailed physiology. And so with these 1,600 patients, we had daily in-depth physiology collected through the 28 days of their ICU or hospital admission. And this data is still sitting there. And there are people like John Sperry at Pittsburgh and a few others. You can get access to this database. And it's great because you can just ask questions. Well, what happens to the patients who had 2 liters of ringers versus no ringers? What happened to the patients that did this or not this? And so this database is really ripe if you're into mining, if you have a biostatistician core that can help you and you're interested in this kind of analysis and just asking questions about how trauma patients respond in different conditions. It's a great database to use. As you see, the entry criteria had to be over 16, just again, IRB logistics. The base deficit or hypotension just meant that they had blunt trauma, they were hypotensive, They had evidence of hypotension with a lactate, and there was a unit of blood started, which meant they were sick enough that some hopefully reasonable doctor taking care of them thought the patient needed blood. Very crude, but it requires easy screening and consistent screening that allowed us to collect this patient population. One of the things we did during the grant, which is what I was in charge of in large part, was developing the standard operating procedures. Again, if you think about it, in the iatrogenic story I told you earlier about saltwater drowning, even though we somewhat superficially take care of patients similarly, if you travel around the country, and if I go on rounds and so forth and actually look at the exact details of what you do, there's a lot of variability in what we do. As far as antibiotics, nutrition, when do you start it, how do you start it? So many different things that we now recognize those things have an impact on the host response. And I can't tell you my way has been proven better than yours, but we're understanding that they do have impacts. And we are undergoing the studies trying to answer which of those variables is correct. So what we tried to do with this is to try to avoid and eliminate as much of that noise as possible. So we did standard operating procedures for ventilator care, antibiotic use, nutritional support, DVT prophylaxis, and so forth, just to get rid of that background noise as much as possible from physician decision-making. And so we did this with 8, 9 trauma centers, and those of you who know trauma surgeons, you know how easily that went. And that's why they gave me the honor of being able to lead this task. So there was a lot of public caning and humiliation and whatever it took to try and get the group to agree. And I make light of it, but it really was difficult because particularly the less data you have, the stronger your bias, the harder it is to get you to change your mind, even though there's zero data supporting what you want to do. And so we fought for 10 years, but we became very close friends over it. And we did, as you can see, we got all the way up to about 40% compliance. And if you look at the sepsis program, what's interesting is you can show a measurable clinical impact when you institute a protocol if you can get any 10% increment. If you go from 10 to 20, 20 to 30, whatever. Even at the low end of compliance, if you just get 80% buy-in, you can start to impact the clinical outcome and consistency of care and decrease in complications. So that's what we did. The NIH is still trying to push people to use our SOPs in these multi-institutional trials because of that, trying to get rid of background noise. Doesn't mean we're absolutely right. It just means we're consistently wrong, and it's not variably wrong. And you can see at the bottom, they were sick. ISS is 32. Third of them got multiple organ failure. They spent 12 days in the ICU and so forth, had a mortality of 16% overall, although I'll show you that over the course of the disease treatment, we went from a 20% mortality when we started, 10 years later it was 10%. And some of that's the standard operating procedures and many, many other things that we changed over that 10 years. But again, argues why you can't use historical controls for these clinical trials. 10 years later, we had a 50% reduction in mortality. And I can't tell you exactly what point it was, but there's all those things we did have an impact on outcome. So we had 167. We looked over 28 days because we wanted to do it serially. And we had to control patients. We did the total leukocytes. We used a genome chip that came out of Affymetrix in California. And then the latest and best way still to deal with this massive data is using ANOVA and EDGE, which I can't even tell you the details of, but it allows for tracking of the changes in a relative fashion back to control of thousands of data points, and then using the k-means clustered method to try and bring the genes that sort of change together and having a similar response over that time period are lumped together more just for visualization and consumption. It doesn't mean absolutely that they're linked functionally. It means that it allows you to track them as potentially linked functional events. And it's a very low probability of false discovery, which you have to have. And what we discovered, and this was one of the big take-home points up here, is that when you get hit by a truck, when you have an ISS of 33, you measure your genomic response and compare it to controls, 80% of your genes change significantly, which is what we nicknamed the genomic storm. But 4 out of every 5 genes. Now, if you think about cancer, you know, if you're lucky with a clean cut cancer, 1 gene can cause it. Or as we're finding with many cancers, 12, 24 genes are mutated and cause the cancer. In this case, out of the 21,000 genes measured, 80% of them change after severe injury. It's an enormous explosion in alteration, which is what we'll go through. If you look at a 1.5-fold difference, then it's down to 10,000 genes. And if you look at a 2-fold change, 5,000 genes change significantly at two times change in their activity level. The other thing, as I said, we don't know. We say 2 times must be important, but I'm not sure evolution established that 2-fold change as being important. I mean, if you have 10,000 changes as at a 0.1-fold, it may mean more than 10 changes at 2 times fold. It's the way we used to look at things, but we really don't have absolute proof that we should be using a 2-fold cutoff or any fold cutoff. It may be just the pattern of change that's important. So this is where the quiz will be after the lecture. So line 30, 23 here, this is K cluster 22. I want you to remember this pattern. So this is a heat map. I think most people are starting to get used to looking at heat maps. Again, it's a visual display that allows you to sort of consume a whole lot of information very simply and easily. And again, it gives you patterns and trends. If you pick it apart and pick one gene and say this is wrong, I can't argue with you. But it's the patterns. And if you look at these patterns, they all do the same thing. Across the top is the day which the sample happened. This left side is control. And these are the 5,000 genes that were 2-fold changed. And these are the control matching genes on the left. And then these are the patients at 1 day out to 28 days. And you can see up here, there's this group of genes that are blue. Blue means downregulation, cold. So in the control patients, these genes are basically turned off and not functioning. As you can see, I quit losing my arrow. After the injury, there's a marked change from blue to red. And then by 28 days, the patients are returning, quote, to normal. And these cells are starting to turn off again, and they're going back to blue, which is where they started before they got hit by the truck. 2 things. One is, at 28 days, you're still not back to normal. And if you look at your severely injured patients, they may go home on day 28, but if they've been in the hospital 28 days, you know they're still not back to normal when you send them home. They got another 6 months of recovery ahead of them. And this data confirms that they aren't back to normal. The other is that you can see the different patterns. The upper part goes from blue to being upregulated. Here, the bottom bottom is red in the control patients, and they get turned blue, which means they get turned off. They're suppressed, their activity suppressed. My whole career, when I told you about it, the beginning paradigm was we've always focused early on the upregulation, the upper third. And if you look at those, they fit right with what we've been studying for the last 30 years. It's the innate immune system pathways, IL-1, IL-6, TNF, various other mediators of innate immunity are the predominant pathways that are turned on, and this is shown schematically in the genomic level. But amazingly, and this is new from this data that broke with our paradigm that I showed you earlier, 2/3 of the changes go from upregulated to downregulated. And if you look at these genes, these are those immune system, again, suppression that I mentioned in that first diagram. But what this shows is it happens immediately. The suppression does not happen in response to the over-response of the innate. And so your adaptive immune response immediately after the injury gets suppressed and turned off. And so there is no suppression in response to overactivation. The aberrancy and the disruption of the genomic response occurs simultaneously. You have upregulation of the innate response, you have organ injury, you have multiple organ failure, and simultaneously you have suppression of the adaptive immune response, and you have your secondary infections, nosocomial pneumonias, ventilator-associated pneumonias, and so forth. And the pattern is consistent. It was first shown in this data set. One thing, just to give you, right here, you see these sort of red ones right there? So that's upregulated. If you look at that small group of genes, it starts out blue. It doesn't become red until day 7. And I can remember from when I was an intern, they used to tell us about this, quote, garbage gamma globulin, IgM and IgG, that turns up at about 7 to 10 days in severely injured patients that, nobody knew what it was for. It didn't seem to be protective. And it was just a aberrant immunoglobulin production, and that's what those are, is immunoglobulin genes. So it's totally consistent with what we had clinically observed. Didn't know when I was an intern what it meant, and today I still don't know what it means. So that's how far I've progressed. But I can show it to you in red and blue on a graft. So those are the kind of things that are confirmed, and also hopefully better target where we need to go with our interventions and therapy. And if you do it on a proteomic level, this is just looking at the cytokine proteins themselves, you can show the same thing. And this just shows that on the top, certain of the pro-inflammatory mediators go up, and then towards the bottom over the 28 days, there's marked suppression of many of the mediators. And again, similar approach, using proteins instead of the genome. So if you start putting these genes together, and there's packages that allow you to look at the genes, and it links the genes that are known to work together. And you may wonder how they have these packages. They hired a bunch of PhD students at $1 a pathway to go through the world's literature. And every time an article was written that these are the toll receptors on the surface of the cell, every time a paper is written that TL4 has been shown to activate NF-kappa B, they put it into the program. And they did that 1 million times. And they just hand extracted all those individual little points of information, put it into a software program. You take the data I showed you, the genes that are upregulated, and the program then links together the genes that have been shown in separate experiments in a cell line or whatever in a laboratory, and they combine those two, and they allow you to put together these activation pathway maps. And if you look at this, these are the TL receptors on the cell surface of macrophages, neutrophils, most immune systems. And you can tell which ones bind with which. There's like TL4 binds with endotoxin, TL7 binds with viruses, and so forth. And you can see that in the patient who has had severe trauma, virtually all of the receptors that activate the innate immune system are upregulated on the cells of these, on the surface membrane of these inflammatory cells. And it just causes this massive inflammatory response through the pathways. The red means that those gene products are upregulated. So you can see that the inflammatory response, the innate system, is massively upregulated by this severe injury. In contrast, this is the network for gamma interferon, basically. And as you can see, again, blue is downregulated. Other than the receptor for IL-1 receptor here, which is upregulated, every other gene in the gamma interferon network is suppressed. And you need gamma interferon to fight infections. And there's been studies done in the past, starting Dr. Hiram Polk had interest in this, because you can see that most of the gamma interferon works through the MHC class antigens in supplementing the adaptive response to infection. And so, once again, we're trying to get a grant to supplement patients who have no gamma interferon activity and supplement them with gamma interferon to see if we can affect their infectious complication rate. Whether it's going to work or not, we don't know, but we will only be giving it to people who need it, and that is people that have this kind of demonstrated shutoff of the gamma interferon pathway, and hopefully we'll be able to block a lot of the secondary infections that turn up. And then just one little other thing that the residents, I'm sure, can recognize, these very sick patients in the ICU frequently have hyperglycemia. You give them lots of insulin, and it doesn't seem like the insulin helps them much. You've seen that kind of patient. This is, again, white cells after a blood transfusion, after injury, the next day. Again, blue is downregulated, and on the top is the insulin receptor on the membrane surface. It is totally shut off. So there's no production, there's no expression of the insulin receptor. And without an insulin receptor, you're not going to respond to insulin. You can give this patient a whole lot of insulin, and you're not going to get much response as far as uptake of glucose by the cells for metabolism. And so again, it's letting us take apart every little component of the response system and hopefully at some point allow us to identify where to attack it and improve it. So this is just look at the upper left. And what this shows is the black at the bottom is the increasing mortality over time. The upper part is the multiple organ failure, which is very high very early on in the course, and then rapidly the multiple organ failure clears. And one of the challenges we had looking at this data is only 15% of the patients died. And so even though we had a fairly good number, we didn't have enough patients dying to really use death as the yes-no endpoint. And so what we came up with was using time to recovery. And so I'll tell you, the endpoint is if you recover from your multiple organ failure in less than 5 days, you're in the good group. If it takes you 2 weeks or more than 10 days to recover from your multiple organ failure, you're in the complicated group. And remember, again, these are all trauma, blunt, hypotensive, very similar patients, and yet 1/3 did very poorly, 1/3 did very well. And we started to break them out based on the time to recovery. And on the left is the innate immune response, and on the top is on the right is the adaptive immune probe sets. And again, as you can see, the innate immunity is pretty much suppressed before the injury becomes activated and then returns towards normal. The adaptive is the opposite. It tends to be upregulated. It's suppressed after injury and then comes back towards normal. And if you look at the cartoons, it's just taking, again, this data and expressing it in little clumps of genes that seem to be moving in the same direction and together and may functionally be related. So on the innate, you have a massive increase in activity, and then the patients that don't have complications rapidly return towards normal. Those that have the complications and spend 2 weeks in the ICU recovering do not homeostase, do not come back towards normal as the other group. The other thing it points out is these are the genes that and the proteins that we've been measuring for the last 20 years, looking for a magic biomarker, saying if your IL-6 is high, it's going to tell you how the patient's going to do. And the problem is, right here is IL-6. And it's the same in the group that does well as in the group that does poorly. And so looking at a single cell or small group of not cell, single protein or small group of proteins doesn't help you. It's the same thing with immunosuppression. You look at genes here, proteins here, in both groups, it's the same change. It's not one goes up and the other goes down, which is what you like with a biomarker. They both are apparent. The difference is one rapidly recovers, the other doesn't, and it correlates with their clinical outcome. And so again, this is just an expansion of those 5,000 looking at the two groups. And again, this is just pulling it out, looking at the various clusters. And again, each cluster actually showed up a similar pattern. If it's suppression or activation, it's the return to normalcy, which is markedly different. And again, different expression. And what it came down to was that if you look at a twofold difference again, assuming that that's more important than a one-fold difference, there's 63 genes that will predict how the patients are going to do. But it's not a single point early on. It's 2 points early on because they start to dissociate very quickly. And we've developed a nanostring technology, it's called, so we can look at just those 63 genes as a pattern. It takes 4 hours to run the test, so it's becoming almost clinically relevant. And we can run those at 2 time points in these severely injured patients, and by the response, we can see where they are on those 2 curves, and we can predict whether they're going to have a very complicated course or a return towards normal. And if you look at it and use it as a predictive model, people like ROCs. I don't particularly like them, but people like them. And so this is like every other trauma score you've ever seen handed to you. And my score, I'll sell to the COT, is much better than anybody else's score. So as you can see up here, this is the Apache ISS NIS microarray Reben, and this is our nanostring. Our 63 genes is the one that gives the best prediction of outcome in the ICU, by far. And so that's what we're trying to convince the government. So over 80% of the genes change, 5,000, two-fold change. That's a lot of genetic stimulation. If you ever want to sit around it in a bar somewhere, think about that. Long-lasting, 28 days are still not back towards normal. And then again, increased our innate, suppressed our adaptive immunity. And then I have a couple more slides I want to show you. Don't leave yet. This is for the rest of you who don't care about trauma. So this is all, we had to use control patients because we didn't have the patients just before they were hit by the truck. So everything's not compared back to the same individual, which is a problem. We use controls and assume that a normal person is the same as a motorcyclist, which has been argued. But that's what we were restricted to with our data sets and our patients. So there's been a couple of little studies done. This is one I want to show you that actually came out of Miami. And they wanted to look in the same patient after surgical imposed trauma, whether they could find similar changes that we found. And so again, this is a predictor. They looked at patients that underwent major thoracoabdominal surgery and see if the insult of a major operation was similar to major blunt trauma and whether they could predict outcome with it. So they took these patients and they sampled their blood just before and 24 hours after either a Whipple or a transhiatal esophagectomy for esophageal cancer. And they did exactly the same gene assessment. They used a twofold difference. They lowered their false predictability a little bit just because they didn't have that many patients and they needed to make their data a little more robust, but it's a little more subject to potential misinterpretation. And they looked at the outcomes, infection, VTE, length of stay, and so forth. And they were able to do it within patient. They measured the same patient before operation and after his operation. And amazingly enough, the new gene chip with more splicing can tell you about 48,000 genes instead of our 24,000. But again, with a major surgical procedure, they could measure some change in 80% of the genome. Subtle changes, but they could find it. But compared to our 5,000 significant change, they found 500 genes that changed significantly. So you can say that a Whipple is 1/10 as impactful as an ISS.